if the atoms interact between themselves, they can generate stable states, which he calls spin glasses, and the spin glasses are magnetic. And this was an example of how you could build an emergent property. So again, the image in a TV screen is a more common example, but in fact, we live in the world filled with emergent properties. Physics, biology, human societies, most of the things we care about are actually emergent properties. Now, if you think back about the brain, you look at the evolution of the nervous system, what nature is doing, particularly with the mammalian lineage, is building brains that have more and more neurons, and these neurons are more and more connected. In fact, in humans, we have probably the most connected brain, particularly for our size. So now, if you're interested in building emergent properties, what do? You build systems with many, many particles, and you connect them all. Because the more interactions, the more emergent properties. So it's very likely that nature is using the nervous system to build emergent properties. In other words, that's the whole point of brains. That's why brains have more than one neuron. So if you think like that, then and you go back to the neuron doctrine, then you could imagine that we've just been doing the wrong experiment all these years. We've been trying to understand something that's emergent. It could be like a TV screen. And we've been looking at the individual pixels. In fact, we've been looking at one pixel, one neuron from one animal, doing a particular behavior, and comparing that with another pixel from another TV screen, another animal, doing maybe different behavior. So it's not surprising that we don't have yet a general theory of brain waves. So now, how can one capture the emergent properties of the neural circuit? So this is an example of a brain slice of the mouse primary visual cortex. Every one of these white dots is a neuron. And just to give you some numbers, the primary visual cortex of mouse has about 180,000 neurons. So if you imagine that this would be like a TV screen, and you want to understand what goes on here, then the solution is to record from every neuron, to see every pixel. And that's how you can appreciate if there are emergent properties there. So how can we do that? Well, you can try to put electrodes, but this is not going to work, because you're going to turn the brain into Swiss cheese. So we need new technology that will enable us to record the activity of every neuron in a neural circuit. Because otherwise, we're never going to understand these emergent properties because we never see them. So this was the idea behind the proposal that we sent to the White House, which was inspired by the Human Genome Project. Human Genome Project was a successful example of a large-scale technology development program which lasted 15 years and built technology to sequence the entire human genome. Something that at the beginning seemed impossible. And this occurred through the collaboration with molecular biologists, scientists that came from the physical sciences, from physics, chemistry, engineering, computation. Through building large teams of people, they broke through these technological challenges and they designed these machines, big genome centers, that enable sequencing the human genome. In fact, that happened ahead of time, even before 15 years. So we proposed to the White House the idea of doing something like this, to design technology to record the activity of every neuron in the neural circuit. And this is something that we call the brain activity map. And with colleagues, some of them coming from the physical sciences, we sent a proposal to the Obama administration. And this proposal became the BRAIN Initiative, which was launched by President Obama in 2013. And it's slated to last 15 years, the total budget of approximately $6 billion. And right now, encompasses the work of 500 laboratories in the US and abroad. In fact, this is an initiative of funding that is not restricted to the US, so it's open to any researcher in the world. So the goals that we proposed was three goals. One, to develop technology, tools to measure every spike from every neuron in the neural circuit so that we can see the entire TV screen. The second goal was to develop tools to be able to manipulate and change the activity of every neuron at will in the neural circuit, because it's not going to be very useful if we can only see things unless we can manipulate the system. And this is necessary both for research and particularly for the clinic. We want to design new therapies for mental or neurological diseases. We're going to have to get in there and reprogram and change those neural circuits. And the final goal was to develop tools to computationally analyze this data, because otherwise we will not be able to see these emerging properties. These emerging properties are not going to look like an image in a TV screen. It's going to be correlations in space and time, maybe very complicated. So we need to have the mathematics, the statistics to be able to actually capture them. The idea was to develop these tools first in very simple animals, just like it happened with the human genome, and little by little graduate to larger and larger brains, ending in the human brain. Maybe not, the goal would be not to measure the activity of every neuron in the human brain, but perhaps measure the activity of every neuron in the cortical area of the human patient. So let me just skip through this. So why should we spend all this money to develop new tools? So there are scientific goals. We need to understand these emerging properties, because otherwise we will not understand how the brain works. The medical goals, it's very likely that many of the mental diseases and most neurological diseases have a pathophysiology, which is actually based on alterations of these emerging properties. And unless you measure the system as a whole, you could be in danger of completely missing the pathophysiology of the disease. And then another reason, and this is the one that Obama used to sell this to the Congress in the US, is that with the generation of these new tools, they will trigger a new industry, just like it happened with the Human Genome Project, which was the birth of biotechnology. In fact, it was demonstrated that for every dollar of funding that went into the Human Genome Project, 15 years later, the economy recovered $124 of benefit. So it was a gigantic investment return for this seed funding of technology development. Obama used the same argument in Congress and said, well, if we invest in tools for neuroscience now, we will develop a new technology. Sometimes you call it neurotechnology. that many years later could provide an entire new area of the economy that could help the U.S. Continue to be the world leader economically. This is an argument that Congress supported. In fact, to this day, the BRAIN Initiative has the full support of both the Republican and the Democratic parties. And even though there's been a change in president, the BRAIN Initiative continues with its funding on schedule. So now to make this, make this happen, to develop methods to record the activity of every neuron in the neural circuit, and to manipulate every neuron, and to be able to decipher those patterns. This is a huge challenge, and it's we need to bring together teams of people that come from many different disciplines, not just neuroscientists or clinicians, but also physicists, for example, people using optics, computer scientists to mine the data, chemists, people coming from nanochemistry, optochemistry, molecular biologists, molecular designers, and also some of the industry, the microchip foundries to build these large-scale nano devices. The BRAIN Initiative, as I told you, starts with this proposal, and then once the White House endorses it, since 2013, has been funded by an act of Congress. So every year, Congress gives money. The budget for next year is expected to be around $500 million. And this funding goes to the federal funding agencies, the NIH, NSF, DARPA, others. And these funding agencies are the ones who deliver the funds to the investigators. So the White House is not involved anymore. This is money that goes from Congress to the funding agencies to the researchers. Private foundations have played a very important role, a critical role in helping coordinate the first steps of the BRAIN Initiative, and they are working also from the sides. And the industry is expected to kick in and start contributing funding to the BRAIN Initiative and help bring the project to completion. So the US BRAIN Initiative triggered similar initiatives in other parts of the world. Started in Japan, China, Korea, in Canada. This is a picture from Australia. We have here the Richards here, who you'll hear very soon. And this was the launching of the Australian BRAIN Initiative. And in Canberra, representatives of the BRAIN initiatives from all over the world, including Europe, that had its own BRAIN initiative that happened more or less at the same time as the US. We signed a memorandum which started the International BRAIN Initiative. So now all these BRAIN projects around the world are coordinated in an umbrella organization, which Linda directs, which is the International BRAIN Initiative. So in this map, we now have to fill up this wide territory, and I'm very glad to be here to try to help crystallize this Latin American BRAIN Initiative. So why do we need an International BRAIN Initiative? Well, there are things that we can do together that we cannot do by ourselves. In fact, this is an emergent property, as you know. And one suggestion that Corey Bartman and I proposed is that you could generate technology just like the astronomers have done that could be shared between many different countries. So this is something that we call the BRAIN Observatory. Imagine an astronomical observatory But instead of being focused on the sky, it's focused to the brain. And this could be a very large instrument, a collection of instruments, maybe as big as this building, that could be run by teams of scientists internationally, just like the observatory, that may enable these goals of the brain initiative of measuring the activity of every neuron in the brain. In an animal, for example, you could bring your animal to this instrument, take all the data, then go back to your lab and analyze the data, just like the astronomers do that. So these instruments could be used to map connections with electron microscopy, like connectomics, or to fabricate a large-scale electrode arrays that have millions of electrodes, or for big lasers that could enable the building of large microscopes that could reconstruct the activity of the brain in 3 dimensions, or big magnets that could have a better resolution than the ones that are available right now, and of course storing all these large quantities of data in big supercomputer centers. So this is one example of the benefits of having the International Brain Initiative. There are many benefits, but this is just one example. And this is something that for a country like Uruguay would be extremely helpful to be able to use instruments that may not be affordable by a single country, but to be shared between many different countries. So, so what is the plan of these types of projects? So our proposal was a 15-year timeline in which we would just escalate the number of neurons that we can record from and we can manipulate, just like it happened with the Human Genome Project. So imagine in 5 years, you could record the activity of 50,000 neurons, 10 years, 1 million neurons, 15 years, you could have entire brains in a behaving animal. What could you do with 50,000 neurons? Well, you could record the activity, for instance, of the embryo of the zebra, which is about a little bit more than 50,000, but it's getting there. So this is some other examples you could do. What would you do with 1 million neurons? Well, you could reconstruct the activity of small invertebrates, Drosophila, the human retina, for example, in vitro, the entire hippocampus of a mouse, or the entire cortex of a very small mammal, like the Etruscan shrews, which have the smallest cortex, and each hemisphere is about 1 million neurons. So you could reconstruct the activity of one entire hemisphere of the cortex of a mammal. And then 15 years, you could then reconstruct the activity of, for instance, the entire cortex in a behavior mouse, or a corticolarian awake primate, or in a human patient, for example. So how are we doing? I'm just going to show you some data from my group. We're building technology to reconstruct the activity of entire neural circuits to explore these emerging properties. And this is an example of one method that you probably have heard of. It's called calcium imaging. And this was developed when I was a student with Larry Katz at Rockefeller University in New York. And we used calcium indicators to stain pieces of cortex. And turns out that every time a neuron has active action potential, there's an increase in intracellular free calcium concentration. That happens because voltage-sensitive calcium channels open. In fact, we got lucky because every neuron, as far as we can tell, every animal has voltage-sensitive calcium channels. And the calcium concentration inside the cell is essentially 0. So if even a single calcium channel open, under ideal conditions, you can detect that if you have a calcium indicator inside the cell. Which means that by using calcium, you can indirectly measure the action potential activity of the neuron. And this is not very impressive because you see the calcium signal is small and slow compared to the action potential signal. But what's important here is this is an optical method, and you can take this measurement in all the neurons at once in a non-invasive fashion. you don't need to put an electrode into every neuron. You can just label the cells with calcium dyes and use microscopy non-invasively to measure the concentration of calcium and detect when the neuron has action potential. So in this movie, you start 500 neurons, 10 minutes in the life of this piece of cortex, of the mouse visual cortex, and the gray dots are the somata of the neurons, and they turn red when they have calcium transit. So again, this is a small part of the mouse primary visual cortex, only 500 neurons of 180,000 neurons. So we're only looking at the corner of that TV screen. But in that corner, we are seeing everything. And we can start using this method to explore if these patterns have repeated spatiotemporal properties suggestive of emergent functional properties. So we've used this method in very simple animals, following the inspiration of the Human Genome Project. In fact, the animals that have the simplest brains in evolution are the cnidarians. This is the tree of life. Cnidarians are animals like corals, jellyfish, sea anemones, and hydrozoans. This is a hydrozoan. And about 750 million years ago, for reasons that we don't understand, the first nervous system evolved. And the first animals that have nervous systems were the cnidarians. A little bit later came our branch of the tree of life, the bilaterians. And this involves all these invertebrates and mammals. We are animals that have bilateral symmetry. Cnidarians have radial symmetry. And then if you go before the cnidarians, animals before them are porifera sponges, and they don't have neurons, they don't have a nervous system. If you want to identify the simplest nervous system, then you have to study cnidarians. And it turns out cnidarians don't even have a brain. The nervous system is distributed through the body. In fact, they don't even have a head. This is before evolution invents the head. So we've used this cnidarian called Hydra, and with calcium imaging, we've been able to label every neuron in the animal and measure optically the activity of every neuron while the animal is behaving. This is a little hydra. They're about 1 centimeter tall, transparent. They live in freshwater ponds and rivers and lakes. And they have about 600 to 1,000 neurons that belong to 12 different subtypes. And they're spread out through the body and the tentacles. So these little white dots are the neurons labeled with calcium indicators. And you can see that when the animal is contracting, you can see the neurons are actually firing. Some of these movies, under ideal experimental conditions, we think we're actually measuring every spike from every neuron. So this is one of the goals of the initiative, that we can achieve that already with very simple animals. We can see the entire TV screen. Now, that doesn't mean that we understand what's playing in that TV screen. That's a little more complicated. And we found that a lot of the activity that you see in Hydra is actually spontaneous. It's not related to the behavior of the animal. So this is one of the big surprises of mapping the activity of this cnidarian. So let's move out from Hydra. So you can imagine that in the next 10 years or so, bigger and bigger animals will be mapped, and we'll be able to measure the activity of every neuron or most neurons in these animals while they behave. People are almost there with their worm and getting very close with this. Now about the zebrafish. Now, if you go to mammals, which is what we care about, this is an example of similar experiments in a living mouse, in a weight-behaving mouse. This is calcium imaging of the primary visual cortex. This is the raw data, and this is the analyzed data. And this is what's happening in the primary visual cortex while the animal is looking at these patterns of bars of light. And if you look at the activity of the visual cortex in this mice, you realize that in this one you can see how groups of neurons are firing together. You see how there are little earthquake where a group of neurons is popping. In fact, neurons don't like to fire individually, they like to fire in a group. And this is actually a direct example, demonstration of an emergent property. So these groups of neurons could be the functional units of the primary visual cortex in the neurons. And you would never see that unless you were measuring the activity of all the neurons. So with these methods, we can measure the activity of these groups of neurons, and then once we know who they are, we can try to stimulate them and see what function they have in the animal. So we've been doing this. This is our dream experiment. You use calcium imaging to measure these groups of neurons. We call them ensembles. People have called them by many different names: tractors, assemblies, bumps, groups. The name doesn't matter, but I think you've seen those in that movie. So now the dream experiment is to stimulate them the same order in which they happen as if you were playing the piano. Imagine that each neuron was a piano key. And now you play the piano, you want to play back the same pattern of activity that you see. So we can do that with small numbers of cells. Again, we're still looking at a very tiny corner of the TV screen of mouse visual cortex. We can use a method, two-photon optogenetics, and combine it with two-photon calcium imaging. So we have a mouse in a microscope. There are two lasers, one to image calcium and the other one to stimulate neurons optogenetically. And that's the way in which we can do this dream experiment of watching groups of neurons firing and then playing them back. So we can do this in an animal that has been trained to lick every time he sees vertical bars. See how the animal is licking after vertical bars. And then that's the go stimulus. Then when we show the animal horizontal bars, the animal is trained not to lick. This is a go, no-go task. By training the animal to lick or no lick, the animal is telling us whether he saw one pattern or the other. Simultaneously, we can measure with calcium imaging the activity of this part of the primary visual cortex and capture these groups of neurons. So the experiment that we've done is, as I'm telling you, we show the vertical stimulus and we measure the group of neurons that is firing with a vertical stimulus, and when the animal licks, then we show the animal a horizontal stimulus, a different group of neurons fires, the animal doesn't lick, and then the critical experiment is this bottom one, where we turn off the TV screen, there's no more visual stimulus, we turn on our piano, our optogenetic laser, and then we stimulate back the neurons that were associated with the stimulus for the animal to lick. What we found is that the animal licks. In other words, we can supplant the visual stimulus by turning on, playing back these groups of neurons that are associated, that were triggered by the visual stimulus. So this experiment demonstrates that not only are there emergent properties, these neurons firing together, but that these are functional neurons, because if we turn them on, we can trigger behavior. Let's move to humans. This is an example of a patient from our colleague John Donoghue, University of Brown, who is paralyzed, paraplegic, and she has 4 electrodes in her motor cortex connected to a brain-computer interface that controls her robotic arm. And by thinking, she's moving this robotic arm, and she's drinking by her own volition for the first time in 20 years. This is possible through a long training in which mental exercises can actually be detected with electrodes by the computer, and little by little use that loop to train the robotic arm. This is what we can do today, recording the activity of 4 neurons. If we can record the activity, look how happy she is, of millions of neurons in the cortex of this woman. And she had robotic arms and robotic legs. She could have maybe a normal life. So what is the brain building in terms of human technology? So I brought an example of a chip. It's a semiconductor chip built out of CMOS, which is about 2 square centimeters, and it's very thin. It's about 100 microns, and it's flexible. And in fact, you can implant it under the skull on top of the cortex. The idea is to use it as a visual prosthesis, to put it in the visual cortex of a blind patient. This chip is wireless, so you can read it out from the outside. And in fact, you have 1 million electrodes to read the activity of the neurons, and 100,000 electrodes to stimulate neurons. So this is not science fiction. This chip is being tested now in monkeys, and should be approved for use in humans in 3 years. With this type of device, you could imagine a blind person. You would get this implant, and the chip could be connected to a camera, and the person could actually use the camera and get the patterns of stimulation of the visual cortex that could be used to see, to detect objects that are in front of the person. And this is not crazy. Visual prostheses that we have right now are doing something like this with very few electrodes, with 100 electrodes. Now we're talking about 100,000 electrodes. So you could also imagine optical devices so that could be applied to human patients. So this image here is an example of an optical setup that we use to take data like the one I showed you in mice. So this is a big laser table with all kinds of complicated and expensive optics. But it is possible to miniaturize these big microscopes using something called spatial light modulators. It's a holographic method. And with them, you can, in principle, build tiny microscopes. You can put it in your pocket, or you can attach to the skull of a person. And they could have many of the capabilities of these microscopes. So it's not crazy to think that in the future, you can take movies like the one I showed you from human patients. You can do, these people could have small little microscopes that can be either implanted in the hospital for a procedure, and you can measure the activity of large numbers of neurons optically without using electrodes. So these are methods to read the activity in humans, either with chips or with optics. How about methods to activate neurons in humans? So let me point out the work of my colleague Roberto Echenique, who works at the University of Buenos Aires, across the Rio de la Plata here, and he is a chemist, and he figured out that you can use ruthenium to build light antennas that are very sensitive and capture photons very efficiently. But when they capture the photons, they break apart. So Echenique thought that you can actually use them to build cage compounds. This is this ruthenium light antenna. You can attach it to glutamate. So when you hit it with a photon, you can break this light antenna and the glutamate is released. So this is a way to optically release glutamate. Uncage glutamate is the term. Ruby glutamate. And then using 2-photon light, you can uncage glutamate only at the top of one neuron and make it fire, and you have single cell precision. So if you move the lasers a few microns away, the neuron doesn't fire. So this means that you can actually play the piano with one finger using this optical and photochemistry. The advantage of this versus optogenetics is that this doesn't require genetics, and in fact, this compound crosses the blood-brain barrier. So you can inject it in the intraperitoneally mouse, and it actually goes through the brain. And it doesn't do anything until we go with light, we turn on the neurons. So you can imagine, this is the kind of experiment that we can do. You find out what the neurons are, and then you can go and stimulate them one by one, as if you were playing the piano. Now, this is something that you could do in humans. In fact, you could do it in humans today. And why would you want to do this in humans? So you can use this to activate or inactivate neurons. So Echenique also built the GABA version of this cage compound. So this is the light antenna with ruthenium, and this is GABA. And you can release GABA with light. And this is an example of an epileptic rat. This is the LFP recording from the surface of the cortex. These are the seizures. And when he turns on the light and releases GABA, you can stop the seizures. So imagine a human patient that had very strong seizures, not respond to treatment, and he can take a pill with one of these cage compounds, goes through the body, through the brain, it doesn't do anything. And then from the outside, you can go in with light and generate specific patterns of light that either turn off or turn on particular neurons in particular part of the brain of the person. So this is an example of how this neurotechnology can actually be used in humans. So just to finish, these methods that are being developed by the Brain Initiative, other initiatives through the world, can be used to effectively read the activity of neural circuits and manipulate the activity of neural circuits. And I'm showing you how these methods are going to be also done in humans. They start to be used in humans already. So we have, and this is fantastic if you want to study patients or cure diseases, but people could use these methods for other purposes that are not medical. For example, this chip that I show you, which has been designed for blind people, imagine that you could put it on a normal person, and you can connect that chip to a camera, which is an infrared camera. So that person would actually see infrared. So you would augment the sensory capabilities of the person. Or imagine that person, you put that chip in the front of the cortex, and you connect that chip to the database of the stock market. And that person would have direct access in and out through a brain-computer interface, like the woman that we saw, except very high bandwidth, and we have access to all this information immediately instead. Have great economic benefits. Or imagine you could put a chip like this here and connect that chip to not just to robotic arms and legs, but all kinds of robotic machinery, and send that soldier out, controlling a whole array of. So a group of us, which are involved in building this technology, got together, a group of 25, and we proposed a set of ethical guidelines for the development of this new technology. We want to ensure that this technology is used for the benefit of mankind. And our group has people that are coming from neuroscience, from the clinic, from bioethics, from the law, representatives from brain initiatives in all over the world. And we all agree that it was very important to have these ethical guidelines, and we propose a series of measures that we call neuro rights. We think this is a human rights issue because when you manipulate brain, you manipulate the minds of people. You read the information from the brain, or you change that to reading information from the mind, changing the mind. So we think that we're dealing with the basic definition of what is it to be a human, and because of that, we think that this should be human rights protection. So our proposal is to add 5 new human rights to the Universal Declaration of Human Rights. I have time later, we can talk about them individually. And in fact, the reason for my trip, I just came from Argentina, where I've been discussing these issues with many, with the government and many social and academic groups of people there. We're also involved with working with Chile, with the Senate of the Republic of Chile, to amend the Constitution of the Republic to put neuro rights protection, human rights protection against the abuses of neurotechnology and also artificial intelligence, which is very related. And with that, I finish with a quote from Sidney Brennan, that progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. And that you think back about neuroscience and Fahal and Sherrington, the reason that they pushed from the neuron doctrine was because their methods. In fact, we can only see as far as our methods. So even if we think they're very clever and very smart and intelligent, have brilliant ideas, at the end of the day, we can only see what our methods show us. So the people that really advance the field are the people that build new methods that enable everyone to see new things. So it's not that we're smarter than the people that came before us, Stale or Cajal. No, we just have better methods. So I encourage you to think of the importance of methods building for the future of neuroscience, and also encourage all of you Latin Americans to try to join this global effort, because this is, again, something that does not depend on a single country. We need to do it everywhere. Thank you. Do you have any questions?
The experiment you show as a go-no-go, you are taking the example of a few neurons, and that is your map, and then you reproduce the map, and you stimulate the neurons and get the same response.
Yes.
But the original map is not the complete map.
No, it's only a little piece of the complete.
So you are always successful on reproducing that. That means that you are triggering it. You are not activating it.
Exactly. That's how we interpret. So Osvaldo is pointing out that the mouse brain has 100 million neurons, and we're actually stimulating very few of them. We can even do that with a group of maybe 10 neurons, but when we trigger the group, the mouse licks. So are those 10 neurons controlling all the behavior of the licking? Probably not. But you can imagine that they are triggering other groups of neurons. It's like a cascade, like a snowball, waterfall, and they are the ones in avalanche, and they eventually move the tongue of the animal.
The thing is, how many times you successful?
Yeah. So if we can trigger that group.
Oh no, in triggering, in getting the, getting the successful experiment.
Yeah. So let me show you.
Not just the right neuron that they will trigger the whole system.
So it's about 60%. Let me show you so it's not all the time. I don't have the data here, but if we can trigger the whole group, about 60% of the time the mouse licks. Otherwise, the mouse never licks. So we go, well, never meaning 10-20% of the time. So we essentially go from random licks to licking most of it.
I was wondering if we move to the brain initiative. The emerging properties you're searching for are most of the psychologists have been looking for. So my question would be, are the psychologists inside the initiative, how are they related to them?
Yes, the brain initiative doesn't look at where you're coming from. Everyone is welcome, and the people, many psychologists are involved in this, particularly developing methods for creating better scanning of humans. The dream is to build a fMRI with single cell resolution.
Yeah, or EEG or something like that, because we don't have something like that.
Yeah, so people are also working on EEG. But the people that are building the methods are actually not really psychologists. They are physicists and experts in these types of devices.
Because I'm worried that we see the hierarchy, but we don't understand how it works.
Yeah.
So maybe a top-down approach. So there we have the method, we see everything.
Yeah.
And we have the clever ideas.
We don't understand it yet, but this is the same thing that happened in Human Genome Project. They did sequence the genome, but that doesn't mean that in fact they still are trying to figure out. They discover all kinds of things that no one had dreamt about before. Once they have the data, so these tools will not solve the brain, but this will be necessary for maybe the next generation of people, other people somewhere, who would have access to these tools, access to the data, and then they'll be able to actually figure out all these principles.
What I was worried was that you need to know what you need to measure, but this requires like a theory in some sense. If you don't know, how do you know it's calcium, not the, I don't know, phosphorylation of some channel that's really marking the activity?
No, I agree. You need to know what you're looking for, but in this case, what we're looking for are these emerging properties. What we're finding in this first step is that there are emerging properties there, and they are functional. So, but of course, this is just the beginning of the journey.
Coming from Queensland University, Brisbane, Australia, and she's involved in the Australian Brain Initiative, and now so you've been in contact with Rafael Scher about one of the longest brain initiatives that Linda is in Australia working now in China. So maybe we can be inspired from those examples and others that are going on now.
Okay. Good morning, everybody. Thank you very much for having me here. It's a very exciting meeting, and I hope that we can share some of the experiences of the Australian Brain Initiative that may help you in considering how to establish latbrain initiative. Before I start talking about the Australian Brain Initiative, which is much more detailed, I thought I would just give an overview of the International Brain Initiative. I'm a spokesperson for the International Brain Initiative, which there are 2 spokespeople and 2 co-chairs. So let me just explain a little bit about the International Brain Initiative, how it came about and who's in it and what does it do. So as Rafa mentioned, there's currently 7 brain initiatives involved in the International Brain Initiative, and they're shown here on this map. And really, the initiatives came together because there was a need for better coordination between the initiatives. So this is very different. It's not like a society. It's not like the International Brain Research Organization or something like this. It's a coordinating group between brain initiatives. So, it's to try to coordinate the efforts across these different brain initiatives. That's the main purpose. And as Rafa mentioned, these brain initiatives came together because each is involved in asking very big questions about how the brain is put together, how the information is encoded and stored and retrieved. And so it was recognised that no single initiative is going to get there. And so given that there was large investments happening across the world, it was felt that bringing those groups together to try to coordinate the efforts may give us even more leverage and better value for our research dollar. So the vision of the International Brain Initiative is to catalyse and advance ethical neuroscience through international collaboration and knowledge sharing by uniting diverse ambitions to expand scientific possibility and disseminating discoveries for the benefit of humanity. So a big kind of vision. And we've developed 4 major aspirational goals, which are shown here. The first is to promote coordination and leadership among scientists in large-scale research programs and to come up with innovative funding solutions. I'm going to talk a bit more about that. Across borders, between countries, between different research fields, as Rafa mentioned, it's very important to bring together cross-disciplinary influences to really make the most of our research efforts and between these global research efforts. One of the major goals is to share and disseminate this knowledge, both to scientists but also to global citizens of the world, to accelerate discovery and to drive translation and application of our discoveries. And then, of course, to also shape the future by being able to have an influence on future generations of neuroscientists. We actually need to begin training more interdisciplinary neuroscientists because, as you've just heard from Rafa, the very different disciplines are coming together in order to be able to analyse these big data sets. We need to have people trained that are very cross-disciplinary, but also trained in how to collaborate in large-scale collaborative efforts as well. And of course, I'm sorry, I have a bit of a sore throat at the moment. To promote brain health in developing countries as well as in across the world, and also in terms of ethics. So the last point that Rafa was making about neuro rights is something deeply embedded within that each brain initiative currently. So to give you a little bit about the history, of course, the US Brain Initiative and the European Human Brain Project were started around 2013. And the Japan Brain Minds Project was also started around 2014. So these are the 3 major funded brain initiatives. Of those 7 that I talked about, the other 4 are what we call emerging brain initiatives, because their governments have not announced the major funding for those initiatives yet. So even the one I'm going to talk about, the Australian Brain Initiative, I can give you an update as to where we are, but we do not have a major brain initiative yet funded in Australia, but we're getting hopefully close. So there were activities really began to catalyse in 2016, and in a meeting that was organised by Rafa and Corey Bargman at the Rockefeller University in New York. This meeting, Coordinating Global Brain Projects, really started to look across the world at where might there be emerging brain initiatives. And in fact, it was a pretty amazing meeting. It was held concurrently at the same time that the UN was meeting in New York. And so in the afternoon of that meeting, there was a link over to the UN General Assembly where there was a commitment made by various people there that brain was something that the UN also wants to focus on. And so this tells you one little thing about how important it is to leverage this kind of meeting to government and to organisations like the United Nations. So the scientists that were organising this meeting over at Rockefeller, they immediately saw the link to be able to kind of promote brain at the UN, which was happening at the same time. So it's thinking about how you can leverage these activities and really advertise what you're doing to both the public and to government. So in this meeting, I think each person got 5 minutes to speak because there were something represented from 50 different organisations. Of those, the ones who were more committed and kept meeting were those 7. And so there were a few more meetings throughout 2016 and in 2017. And at this point, this Global Neuroethics Summit was started by the US Brain Initiative, and it is funded by the National Science Foundation in the US mostly, but also by the Kavli Foundation. This is this public-private partnership. And they immediately, through efforts of scientists such as Rafa, have really started to push this coordination of neuro rights and ethics within the different brain projects. But that one really started from the US Brain Initiative and the European Human Brain Project. So this culminated in this meeting, which Rafa just mentioned, which we held at the Australian Academy of Science in Canberra, and brought together the representatives from the different brain initiatives. And it was an amazing meeting because throughout the day, each person, we had been meeting for almost 18 months by this point, and at each meeting, what had happened was the different people representing the brain initiatives were all trying to understand, what are you doing? What are you doing? What are you doing? What are you doing? And that was kind of the reason to meet. But it wasn't until this meeting where each person stood up, started giving an update of their brain initiative, where they said, what we really need is to be able to work with X or to be able to work with Y. And by the end of the day, in fact, this happened in the afternoon tea break, we decided to write a declaration of intent to form an international brain initiative, because we kind of had been working around the edges of this, but until we had a declaration, something written down that we would actually work together, this allowed us to draw a line in that and now figure out how we were going to work together, not that we should work together. So that was the Declaration of Intent, which is on the IBI website. And then from there, this allowed us to start to develop those aspirational goals, our vision So we are going to form an international brain initiative. What is it going to do? What's the vision of it? How is it going to be different from IBRO or the SFN or the Uruguay Society for Neuroscience? What is it going to do that's different? And how can it add value to the existing brain initiatives? And so these were meetings that happened during 2018, and we started to think about how will we operationalize this? How can we actually get things done? So we started to bring forward the concept of different working groups. And by this time, this global neuroethics working group had already started to really take off, and because it had funding from NSF and the Kaplan Foundation, this became basically the first working group of the International Brain Initiative. And then we started to develop other working groups I'll talk about in a moment. Then we started to develop the organisational structure in 2018, and throughout the rest of 2018 and into 2019, it's slow going because you can imagine many different viewpoints, different cultures. So it does take time, but it's worth it. And I have to say, the people involved are very, very good to work with. They're very respectful of different viewpoints. And people do science somewhat differently in different countries and have different priorities. So this requires negotiation and consensus. So this is what has been happening, and now we have developed a couple of extra things, which I'll talk about, the Funders Collective and a Stakeholders Collective. And just in, when was that? June this year, we held a strategy committee meeting and this is some of the people that were there. It was a very small meeting to really now start to accelerate how we operationalise the RBI. So you can imagine it's like establishing an organisation of a completely new type that hasn't existed before. You can take some learnings from how other organisations are set up, but it essentially is trying to do something pretty different. So how do we make possible the exchange of ideas? How are we actually going to do it? It's nice to say we want to do that, but how are we actually going to do that? How are we going to bring forward more opportunities and challenges? How are we going to amplify the impact? And how are we going to build these collaborative networks? Something that really has emerged within this group is the need for new ways of bringing together government, industry, scientists, and the public, and funders together, pulling behind this single cause to understand the brain. So, this is happens in different pockets. So there are industry bodies, there's philanthropic organisations and government funders. And of course, there's scientists and there's wonderful scientific societies. But having focused groups that involve all those different people coming together is more rare. And we believe that this will augment these national brain initiatives and help to leverage the resources from each of those. So the structure currently is this strategy committee, and the strategy committee has 2 people from each, 1 to 2 people from each of the brain initiatives involved. And that's the strategy committee. The co-chairs of that are Shigeo Okabe, who's the director of the Japan Brain Minds Project, and Jan Bialy, who's the Deputy Director of the Science for the Human Brain Project in Europe. So that's the Strategy Committee. We have developed ideas for 6 working groups. and not all of them are actually up and running yet. So we do have the Global Neuroethics Working Group up and running. We have the Inventory of Brain Initiatives up and running. I'm going to talk a little bit more in detail about each of these working groups in a minute. The Data Standards and Sharing Working Group is going to meet for the first time in January 2020, so still in the stages of setting up. Communication and Outreach Working Group is currently up and running, and Education and Training is still getting going. This one, Tools and Technology, is still, we don't have the co-chairs of that yet. So here are these working groups. Let me just tell you a little bit about them in more detail. So these are the co-chairs of the current working groups. As I say, we don't have really co-chairs for tool and technology dissemination, although Yves de Koning, who's the chair of the Canadian Brain Research Strategy, is probably pushing this working group forward. So in terms of communication and outreach, this is, so the Human Brain Project, Alastair Thompson is their communications person from the Human Brain Project, and the International Neuroinformatics Coordinating Facility has provided in-kind support for this. So this is another point to make about the brain initiatives. At this point, we have not thrown in funding together. Each brain initiative has its own government funding, and so they provide in-kind support. So, for example, this is a good example. So the HBP have said, Alastair, who works for us, he can also have some of his time to work for the International Brain Initiative to help get things started. So that's communication and outreach. So they run the website, and they also put out press releases, which is another good thing to think about. This meeting, you could have a press release that goes out to all of your countries about this meeting. What that does is to help raise the profile that you're starting to generate a coordinating group starting a Latin Brain Initiative. So these are things you're not trained as a scientist to do, right? But this is what's necessary in order to get government support. So perhaps I'll talk about this one next, because this one is probably the next farthest along in terms of the working groups. So it became evident from that Rockefeller meeting that there needed to be some inventory of what was happening around the world. And so there have been a couple of meetings, and the co-chairs here are Amy Bernard from the Allen Institute for Brain Science and Stephanie Alvin, who's from the Kavli Foundation. And essentially what they're developing is, and they're probably going to use some commercial software for this, to scour the internet to look for projects that are currently funded under the US Brain Initiative and the Human Brain Project. Those 500 investigators there, you'll be able to look up who's got what kind of projects under the different brain initiatives. And also, actually on the US Brain Initiative website, there are funding opportunities that are on that website as well. And this inventory will also try to pull those together as well. And part of that is very important for the funders. So it might be important for you as a scientist to know, is there another scientist somewhere working on this? But it's very important for funders to know, because they want to know, am I funding something that's quite unique, or am I funding something that's part of a bigger picture? Who's working on this around the world? So actually the funders are very keen on having this inventory. So that's that one. And I mentioned Global Neuroethics, who've had 2 meetings, and they have their 3rd meeting coming up at the IBRO World Congress, back to back with the IBRO World Congress in Korea in Daegu next month. They just had a special issue of Neuron in January that came out, and each of the brain initiatives wrote an article for that special issue on the ethics that they are, how they're incorporating neuro rights and neuroethics in their brain initiatives. So you can have a look at that. With regard to the education and training, so IBRO already does a lot of education and training around the world. So we don't want to compete with that. We want to incorporate that. So Tasya Asakawa, who is from IBRO, is one of the co-chairs, along with Sung-Jung Jong from the Korean Brain Research Initiative. So Ginny, who's also involved in the neuroethics, global neuroethics group, has a passion in this area of education and training. And this working group hasn't really developed its plan for what it's going to be doing, but here is how we imagine the International Brain Initiative will try and influence the development of training of interdisciplinary scientists in the future. And of course, we have had some interest also from the HFSP in perhaps partnering here as well. Now, the next group that I'd like to talk about is the data standards and sharing. So imagine that you have taken on Rafa's calcium imaging work, and you've decided that you wanted to work in a South American animal, and you've got fantastic data. You want to be able to share that data, and we need standards in order to understand what file type, I mean, even the details of how you're going to collect that data, where are you going to put it, so that you can get even more value for that data, and perhaps be able to combine that data with another group somewhere else in the world who may have similar data but on a different species. So this will allow you to have so much more power if we have a more coordinated standard for data sharing. That doesn't mean that, you know, there's a regulation that everybody has to use this method, but some coordination around the way data is acquired across all different areas of neuroscience is the goal of this data standards and sharing. So you can imagine that is a huge problem. In the field of MRI, I would say because of the Human Brain Mapping Project, that has come a very, very long way. And there are formats in which the files can be stored, like the new BITS format, which allows different programs to access that information. But we don't have the same kind of thing for cognitive neuroscience data, or in psychology you were mentioning before, or even for electrophysiology. There are some different programs, but they're not universally used. And there's not a good format for sharing that kind of data. Even in terms of microscopy data, there's an open microscopy platform. Maybe some of you are using that, but these maybe not, in which case, do you know about the open microscopy platform? Maybe this is, you know, this would be very useful for you to know about. So the International Brain Initiative is not, at this point, going to fund or to have somewhere in the world a huge computer farm where all that data is going to be stored. That's not the goal. The goal is to just begin the dialogue around how you develop data standards and sharing. So they're the working groups that we have at the moment. There is a process for putting forward a working group, and we've been developing how does somebody come and say, we want to put forward this working group. So we do have a method for doing that. And there's a lot of information on the website, so I encourage you to go to the website and you can subscribe to the newsletter to keep up to date with what's happening there. So that was all I was going to say about the International Brain Initiative, just to give you a bit of an overview, because it's still, as you can see, in the very early stages. Even though we've been going for a couple of years, it's, you know, slow going to develop all of these things. I was going to switch now and talk about the Australian Brain Initiative. Not sure, I might have to unplug this. I wanted to show you a little video that's from our website. I don't know if you'll be able to hear it very well.
It's not connected now.
I just unplugged it so that I could. Yeah.
Oh, yes, the
Yeah. Have you seen it?
The human brain, the most sophisticated machine we know of, and cracking its code is the most complex problem science has ever encountered.
It really drills down to the essence of what it is to be human.
We think it's the most complex thing in the universe. Every time we peel back a layer, we see more complexity.
That complexity makes it a challenge to cure disease, treat disorders, and prevent injuries. Survival rates for brain cancer have not improved in the last 3 decades. Dementia can be detected but not prevented or cured. Progress in treating mental illness has been frustratingly slow.
It's actually heart-wrenching to watch how patients and families trying to cope from day to day, from minute to minute. And it's hard not to take on all that emotion yourself.
But thanks to solid research and recent advances in technology, the horizon of possibility has shifted. The answers to fundamental questions are within reach.
I think it's a very exciting time to be in brain research. We can do things that we really didn't dream of before.
The next few decades will be crucial. Brain science is becoming big science.
Science is the way forward. It's the only way we're going to make a difference to these terrible diseases of the brain, be they epilepsy, Parkinson's, stroke, no matter what they are. The way forward is science.
A national brain initiative would exploit our unique research strengths, raising the prospect of breakthrough therapies for thousands of Australians.
Australian science punches above its weight, particularly in the area of brain science. This would further supercharge it.
The holy grail that I would love to crack is the answer to my patients' diseases, how I could stop them having such severe diseases even before they start.
But brain research is also driving technology forward. Cracking the brain's code puts the future in our grasp, and Australia can lead the revolution by transforming lives. We showed it was possible with the cochlear implant.
3 years old, but never heard his father before.
Talk to him, Daddy. Daddy loves you.
And we can do it again.
We have the potential in terms of the people, the technology and the ideas to really take things further.
The structures of the brain are also inspiring next-generation computers. We're on the verge of the scientific breakthroughs of our lifetime, curing disease and creating advanced technologies. We're beginning to crack the brain's code, and you can be part of it. Become a brain champion today at brainscode.org.au.
Okay, so it's a public outreach video directed specifically to our politicians. So it's very Australian focused. And so I just wanted to switch now and talk about the Australian Brain Initiative and where we're at. And this may be more useful in terms of sort of strategies for developing your own brain initiative. So in those years of 2017 and 2018, I was attending their meetings of the other brain initiatives, trying to understand what does Australia have that could contribute in a maybe not totally unique, because everybody wants to understand the neural basis of cognition. I could tell you every single brain initiative, every neuroscientist wants to do this. But are there things that might be, some evidence that we could have from Australia that may be something that we could contribute a little bit more? So I'll just run through sort of what we have come up with as an example. And I should say at this point that every single brain initiative has come about in a different way. So there's no formula for how you develop a brain initiative. As you heard from Rafa about the US Brain Initiative, you'll be familiar with the European Human Brain Project came about because the European Commission had a huge funding opportunity for missions, big, bold missions. And they put forward a project proposal, and that was funded. So the Human Brain Project is actually a grant from the European Commission. And they've had their first 5 years, and they're now in their second 5 years of that grant, which was 1 billion euros over that 10-year period. The Japan Brain Minds Initiative similarly came about through some campaigning, but also a grant. So in Australia, we don't have this kind of big grant mission mechanism. So instead, we've had to go about it in a different way. And I know that in Korea, it's a very similar process, and also in Canada. In China, they have been developing their brain initiative for some years, and it's still not actually announced yet. And my interpretation of this, maybe you have a different interpretation, is that it's a slightly different way that they've gone about it. And there isn't a right way or a wrong way here. Really, from the US Brain Initiative perspective, my understanding is more that there was the conceptual idea, which was put forward to the government, and President Obama gave a mandate which allowed funding to start to happen. But and there was the conceptual idea that it would be neurotechnologies, but exactly what projects were going to be funded was not all mapped out in advance, right? In China, they are mapping out in very great detail what is going to be funded before they announce the funding. So it's kind of a bit different. And so with the Australian Brain Initiative, we don't have this big mission mechanism. We have to put forward a case to the government to fund a brain initiative. There are precedents in Australia. So the Australian government last year committed $500 million to something called the Personalised Medicine Initiative. And this is to take genomics and to get it into our healthcare system. We have a public healthcare system in Australia, and so this is to develop ways of getting genomics into that healthcare system. So this is a campaign to get the government on board to provide us with the funding for a coordinated brain initiative. So it's a little bit different, and I can now tell you about sort of how we're going about it. So the Brain Alliance are the groups of people that have come together to push for a brain initiative. Okay, so that's the two terms. So these are the groups from around Australia. So we've got scientific societies, the Australasian Neuroscience Society, the Australian Psychological Society, and then a bunch of universities and some companies that are involved. And all of this sits under the Australian Academy of Science. So it's something so the Academy people are not here yet, is that right? It's something to consider. So around the time when we started this, I was president of the Australasian Neuroscience Society and considered whether it should be something the society pushes forward. However, we decided to go with the Academy for very specific, two specific reasons. Firstly, the Academy is a science policy kind of organisation in Australia. They do science advocacy and they do government science policy liaison. So they have a lot of contacts with the government. So we could use that to our advantage. And the government sees, so the Academy is like the Royal Society in the UK, and I'm sure you have the same here. It's a learned academy, so highly respected, and the government respects that. So this is the peak body for all science in Australia. So that's the first reason. The second reason is that it allowed the Brain Alliance to include many more different groups than would be in just the Australasian Neuroscience Society, which has a similar kind of membership as a very, very small SFA, although not as much cognitive neuroscience as we would like. Whereas this allowed us to really bring in the cognitive neuroscience societies, their neural engineering groups, their computational neuroscientists, etc., etc. I guess one other thing about the Academy is that they have other groups, so they've got the physicists organising their decadal plan for physics for Australia. They've got the mathematicians organising their decadal plan. What are they going to do in maths for Australia in the next 10 years? So being under the Academy allows us to liaise immediately with those other groups as well. So it's one way to go about it. But I don't think any of the other brain initiatives have their initiative under their Academy. So just something that we did. Right. Now, how is it funded? So at the moment, each of these organisations pay a membership fee to the Academy. And that membership fee pays for some staff to help us organise things. And I'll show you at the end, we're developing a business case and we needed to get some external help with developing the business case. So that money also pays for the business case. And so they just pay their membership to the Academy and the Academy then pays all the bills for us. And how did we get it under the Academy? We had to convince the Academy to take on brain science as one of their flagship areas of science that they want to advocate for. So they advocate for astronomy, they advocate for geology, and we pushed for them to advocate for brain science, and they agreed to take it on. And that's when we had this meeting, which Rafa came to, and we signed the declaration for the International Brain Initiative. This was so good for us because the president of the Academy was there, and he got very excited and said, he was all for it. So then, it helps to build that support. So this is just our history. We started with there was a committee that then started to put together this brain initiative in 2016. We developed this membership model and we wrote a paper which came from that summit at Rockefeller. Some groups were invited to put together a paper. This helped us to also focus what was the Australian Brain Initiative going to work on. And then following that, this is what we call the Brains at the Dome. The Shine Dome is the home of the Australian Academy, and that's where we have the declaration. Now, on here, there are a couple of things to note. We still don't have funding, and we started this early 2016. So it takes time. It takes a lot of time. So if you think that you will start this and you'll get, if it's the same as us, and I think it's the same in Canada, it's the same in Korea, it's the same in China. So the emerging brain initiatives have all been going around the same timeline. It probably takes 5 years to get the funding. So it's not like you can start something. I mean, maybe you'll be lucky. I hope so. But you have to kind of think in the long term that this is actually going to take a while. And it takes a multi-pronged strategy. So we've got this event, which we're going to run again next month. We call it Brains on the Hill. And the hill is where our federal parliament sits up on a hill in Canberra. So last year we had an event inside Parliament House. You can hire it. So again, we had to have money for that. And we made appointments with as many parliamentarians as we could. And we had teams of 3 people go and speak to those parliamentarians. And that movie I showed you at the start, we had that in a portable video booklet, a disposable booklet actually. So we could give it to them, they could watch it, and then we could talk to them about the brain, funding a brain initiative. Now, another that we have is called the Brain Champions Campaign. So this is for the public. And what we ask them to do is to sign up to become a brain champion. And when they do that, so they go to our website, they click on it, and they just have to enter their name and their postcode. It's called the zip code in the US. I don't know what it's called here, but the postcode. And the postcode is important for us because we can match that with an electoral region, so that if we go and see a politician, we can say, we've got 5,000 brain champions in your electorate that support this initiative. So like to leverage it back onto them, because honestly, that's all they care about is being re-elected sometimes. So you've got to think about the way they think. Now, another thing that's happened, I mean, I don't know if you keep up with Australian politics, but we've had 5 prime ministers in the last 5 years. There's been a massive changeover.
We will take over.
Yeah. There's been a massive changeover of politicians. And I personally, it's so frustrating. Again, another reason it takes time, because you'll build it's about the relationship. So you build a relationship with one minister and then suddenly they're gone because a new prime minister comes in, reshuffles their whole cabinet, and then you have to start over again. So perhaps this is another reason it's taken us a bit of time. But I was getting here very close with our federal health minister. So really what we need is one of the major ministers to champion this to the Prime Minister's Council, and then to get it into the federal budget. So hopefully you have somebody who's savvy about politics and can, but actually the Academy helped us sort of map this out. And because they actually helped that precision medicine $500 million initiative, the Academy helped to get that. So they know what they're doing. I'm hoping they know what they're doing anyway. Here I was getting somewhere with that minister, and then there was a change of PM and called an election, and so everything shuts down. They go into caretaker mode. Anyway, luckily that minister actually retained his portfolio, got re-elected, and so I have gone back into him, and he is going to help champion us to the Prime Minister's Council. So we made one more little step. And then we're going back to do Brains on the Hill too. So this is again, we're going to go and see more politicians. And when we did this one, we had a display in the inside Parliament House of about 20 different labs or neurotechnologies that they were working on. So how do you figure out what you're going to work on? Do we still have enough time? Maybe I should stop. Yeah. That's okay.
I mean, a couple of minutes.
All right, so just quickly try to finish up then. How do you figure out what you're going to work on? So we met for a year and debated what would we want to work on. And we came up with this cracking the brains code as the tagline that we want to use. And there was sort of 6 different areas related to the code. And this is a paper that came out in Neuron in 2016. And since then, we've really focused down on technology development. And part of the reason is because of Cochlear. The Cochlear implant was fully developed in Australia, manufactured, and all of that came from basic research in Australia. So it's the best example that we can take to government for how we can take brain research and turn it into a technology that's of great health benefit, but also great economic benefit and to provide jobs. So, so the Cochlear is one of the reasons we've gone for technologies. And so we've mapped out, well, what kind of technologies are being developed in Australia at the moment. There's this one, which is called the Stentrode. I can go through these in more detail later. We have a bionic eye. We have a system for pain. We have a scanning ultrasound technology, which is for treating Alzheimer's disease, another sort of bionic eye, and this epilepsy seizure prediction stuff. We are also pushing technology not only just in the area of health, of course, but into other areas. So the neuromorphic chip, this is a camera technology based on neuroscience and also sensors. Okay, so I won't go through that. This is our brain champions and brains on the hill. This is, you know, the kinds of wacky things you have to do. We do also have an early and mid-career researcher network. This has been very important to make sure that we engage. So what you're trying to do is engage your neuroscience community, keep everybody on board at the same time as pushing forward into a direction that politicians will be able to understand. And they had their own strategic plan. And then just finally, what we have had to do is to develop a business case. So a business case is where you put forward the economic case for investing in brain research. If only we could just go and say, this is what we need to do. We need to record from every neuron. Our government wants to know, what are we going to get back for our money? And we can say, oh, the Human Genome Project, you know, returned huge amounts on investment. And we do use the Human Genome Project as an example, but they want concrete, much more concrete numbers. So we have had to engage and we can't provide that. We have had to engage a company to help us that's kind of respected, to give us that the government will listen to. And these are just some of the pages from our business case. But this one I think is pretty telling, and you're blank like us, that there's no neurotechnology cluster in this area. And these clusters that are around the world, they are they're the areas that are recognised in the world for science, neuro-related science and engineering applications. Even though we have Cochlear, we're not recognised as one of these clusters. And so this is how we're defining technologies, both devices, diagnostics, and neuropharmaceuticals. Yeah. So then the economic benefits come from some direct numbers. So this is American. You can see here's where the US Brain Initiative was launched, and the revenue from diagnostics and devices has really increased. So this return of 9%, that's huge. That's huge. And currently this neurotechnology market is worth 53 billion US. And you need a mix of small, medium enterprises, big companies, and new startups to make this work. This is just Cochlear. So Cochlear was given a grant in '94 of 100 million, and they've had year on year 12% growth. They're now worth 1.3 billion. And this is sort of our projection. So if we remain at this, in 2030, we will have a 2.5% year-on-year growth. With a $500 million investment here, we think that we can boost this actually to here. So the kind of numbers that you need to show. And what are we actually going to do? This is some of the things we want to do. We have 3 areas. We want to leverage shared infrastructure, local workforce development, and coordinated governance. So this is our ethics program. Every brain initiative has an ethics program. We have some programs for interdisciplinary fellowships, also in computational neuroscience and AI. But we also need some shared infrastructure. We actually need a brain bank, so companies want that as scientists. We need a foundry that's a coordinated sort of funded thing. And I think that's it. So this is our ask. Per year, we want 500 million over 5 years. And I think that isn't necessary. These are just some of the companies that we have that we think we can really help them to move forward as well. And what we're asking from the government right now is that we want some small amount of funding, maybe $1 million, to further develop a full business case and then a commitment that they will provide that funding. So that is where we are at, and hope that's helpful.
I think it's very inspiring for us to have an example of how is the process that we have to think about trying to make this initiative, our initiative. So thank you very much.
You're welcome.
So now we have a small coffee break, and I want to connect with some people abroad to see you later.
Very good. There is one here, there and here.
¿Tenés capacidad ahí para conectar? No hay. Sí, tiene que haber. Sí hay conecta ahí. Bueno, cuando alguien desconecte. No, pero ese por ejemplo es igual que el mío, donde está la y ese, sí, creo que es igual, ¿no? Y este. Yo vi uno ayer. No, yo vi uno ayer. Vamos a ver.
Sorry, I was thinking
mean, the students are leaving.
Yeah.
We are losing all our.
Everybody here, I didn't realize that the functions were also. I knew there
No,
was
it's not
happening.
coming. It's just the inflation. It's just the dilution. I mean,
No, porque de hecho ahora
We were just talking about but if people work, to put these things together.
you know,
you have to organize.
we had
Vamos a ponerle tu nombre y contacto.
people they were just training.
Already
So that day, what day the president was in the name of the elected